I told my class enzymes lower activation energy. They still didn't react.
Here is to 3.1 enzymes. Note disclaimer.
In this unit, you're going to be talking a lot about enzymes, but I want you to remember what you learned earlier in Unit 1 when you're talking about enzymes. Remember, the building block of what we know as proteins are made up of amino acids. Those amino acids are very much building blocks to create or formulate what we know as polypeptides or polypeptide chains, which have specific functionalities as proteins. And one of those functionalities includes an enzyme. So this is important to note, especially if you get a multiple choice question or an FRQ, to relay back to that when we're talking about enzymes, we're really talking about connections between amino acids. And remember what their structure is, because this might be important when you receive some sort of question. To further dive in, we talked about the fact that they have different structures. Remember, we go from primary into quaternary. And while you don't need to know this in full detail for this unit, it's important to note that you can fold in a 3D structure known as the tertiary structure, which is really important for eventually creating the functionality. So remember that when you're thinking about enzymes and this understanding that they're made up of amino acids, but eventually come into the quaternary structure, which are a lot of polypeptides together, but were originally based on this idea of folding once we get to the tertiary structure. So what I mean by that is primary structures make up secondary structures, which make up tertiary structures, which eventually make up quaternary structures. And it starts off with that building block known as amino acids. Eventually, they form a multitude of polypeptide subunits and formulate the functionality of that specific enzyme.
Enzyme time!
Let's talk about that little video clip there. You had two individuals. There was one male and there was one female. And these two individuals were on the bottom of the hill. I want you to think of that as your reactants in a chemical reaction. So let's represent that with the letter R. And the arrow is there, which means the direction in which the child moved up the hill. And you notice that he turned on this little battery pack, which was considered to be an enzyme. So I'm going to represent that with the letter E. And he was able to get up that hill with such ease and complete his journey. And he was happy because he was able to do this with more energy and, well, not more energy, more quickly, I should say, and less energy. He himself did not need to exert as much energy, and he was able to swiftly go up that hill more quickly. And that is what an enzyme is. Yes, we learned that enzymes are proteins and they act as what we know as catalysts. So when you have a chemical reaction, this means that they accelerate that behavior of a chemical reaction. So for example, in Unit 1, we talked about how you could have some hydrogens and they could be added to some oxygens, and eventually you get something like H2O or water. So you have some oxygen and some water that eventually makes water, or hydrogen and some oxygen that makes water. So those are your reactants and those are your products. Please note that this reaction is not perfect. It's not balanced. If you've taken chemistry before, I'm just letting you know in general you have some hydrogen, some oxygen in your reactants, and they eventually combine together to make water. In this whole reaction, what ends up happening is if you had an enzyme, the behavior of acting as combining hydrogen and oxygen to get your final product would actually act more quickly and less energy with the assistance of an enzyme. So you would have more of those hydrogens and those oxygens combining together to eventually make water. And that's what they mean by accelerating chemical reactions. Yes, we would still have hydrogen and oxygen that can combine together to make water, regardless of whether or not there's an enzyme or not. However, the enzyme is beneficial because it allows it to go more quickly and requires less energy. Without the enzyme itself, yes, water will still be formulated, but it takes longer and demands more of an activation energy, which we're going to talk about in just a second. The goal is to basically ensure that we can create the final products with such ease, and that without the enzyme itself, it would just occur at a slower pace, which would just not be sustainable in a natural world. So our bodily functions would not be able to react normally without an enzyme because of the timing that would be exuded. So let's talk about this picture over here, which talks about the relationship between the energy and the reaction coordinate. So you have some reactants here, which has glucose, C6H12O6, and some oxygen, and then they produce products of CO2 and water. This reaction can occur without an enzyme. It just will take a lot of time to go up that hill to eventually get to your product. So we say the activation energy, or the energy that is required for a reactant to go to a product, is a lot when we compare it to with an enzyme. So if we have an enzyme, you could see here the reaction looks like it doesn't take that much time to go from a reactant to product. And that's because our activation energy is a lot smaller. So you can see in this graph here, the activation energy without an enzyme is a lot bigger than when we compare the activation energy when we're utilizing the enzyme. The overall energy that's being released is going to be thought of as being going from reactant to product regardless. So it doesn't matter with or without an enzyme what that really matters. Our main focus here is to note the activation energy. So the goal of an enzyme is to lower that activation energy, which allows the reaction to occur much faster and far more efficiently. So that's an overall process that you see there when we're talking about enzymes. Now, let's talk about how enzymes really form. So there's a couple of terms that you need to note when we're talking about enzymes. This is our enzyme here. So as you can see here, the enzyme looks like it's a specific shape. And we have what are called substrates. And the enzyme has a certain location where the substrates have to attach to, and that is known as the active site. So eventually what happens at the active site is when the substrate is going to attach to the enzyme itself, and then is called the enzyme substrate complex. The structure is really important to note because it's essential for the enzyme's function. Remember, we talked about primary, secondary, tertiary, and quaternary. And if you remember in Unit 1 and in Unit now, we're talked about tertiary structure in that 3D fold of an enzyme because that's really crucial. The 3D structure of an enzyme is really important because it allows it to function properly. This temporary structure of an enzyme substrate complex eventually will break down. So you still have the enzyme there, and then the products, which is taken from the substrate to the products, you can see there, it's the same thing. It's just altered in that enzyme substrate complex, is finally formulated and released from the enzyme. So the structure for the enzyme is really important because if you experience any sort of alterations, which we're going to cover in a later section, such as temperatures or pH, the problem is the enzyme will be less effective. And this is a phenomenon known as denaturing. So if you have something like pH or if you have something like temperature, that's an important factor for the enzyme substrate complex, it won't work properly. So what I mean by that is, let's say, for example, we say that a specific enzyme, we're going to call it E, needs to be able to work at a pH of 3. And if you've taken any sort of chemistry class, you'll know that's a very acidic environment. So let's say, for example, you have your stomach and there are certain enzymes that work in a very acidic environment. And say, for example, there is a enzyme that's in the stomach where all of a sudden the pH now changed to 8. And that would be a problem, because that means that enzyme substrate complex is not going to correctly utilize the enzyme and the substrate to eventually produce the proper products. It needs to be optimal. So that means that we need the right conditions. Environmentally, the pH or the temperature, etc., needs to be correct for it to maximize the production of products. This is an important factor to understand, and you might see some questions related to this. But please note that if you're given a question like this, we're going to be covering this in section 3.2 and even later on. But just remember, when you think of optimal conditions, think of the idea that enzymes are involved. This is an important key function to understand. Enzymes can also, once they're being released, so once you have the products that are released, please note that this needs to be prepared to go back to start to do the process again. So we could start to use the same enzyme again and again and again, as long as there's enough substrate for it to attach to and produce enough product. So please note that this is important. Also, when you see the term 3D, think of tertiary and think about how that enzyme substrate complex is important. So that way the correct molecule acts on the correct enzyme and can produce the correct products when it's being released from that enzyme substrate complex. This can also help you out when you're talking about reusability of that enzyme. So this is important to note overall. Now that we've covered this, let's talk about this idea of the induced fit mechanism. The induced fit mechanism is the mechanism that we now consider to be true. It's this idea of how the enzyme changes shape when binding to that substrate. So this process right here, what happens when the substrate attaches to the enzyme, it changes the overall shape. You can see that together it formulates this little hoop and bubble. So there's a very specific shape that's being formed. And the active site here is very adaptable, meaning that whatever substrate comes and attaches to the enzyme, it will attach properly for better stability. This stabilization is important because it helps assist in the process of being a really great catalyst. And what's also important to note is that you could have the same enzyme that binds to different types of substrates. So you have substrate 1 or substrate 2 or 3 or 4, and that can happen. And the thing that it's important to note is this induced fit model basically says that it's whatever enzyme is best fit with whatever substrate at that time is going to be most efficient. And it's a rotating cycle, right? So it's going to happen again and again and again. And that's what we consider the induced fit model. Now, when we talk about the active site and the substrate interaction, what was previously known, and you might see this in old textbooks, is the lock and key method, which basically says that you have some active site and that the key fits directly in that active site, and it fits perfectly, and that it's not going to be flexible for other substrates to go in. And we now consider that to be false. We now know that an enzyme has a shape specifically at the active site for a substrate, and once it's secure enough, it will maintain a sense of flexibility to retain the specific need of that substrate to eventually produce a product. And so that's why that is important to note.
I liked you better when you changed for me. Yeah. Induced fit.
I'm going to preview some information that you can see later on in this unit, but I want to bring it up early so you understand these terms. Remember that we said a substrate usually attaches to an enzyme, but sometimes you can have issues or problems known as competitive inhibitors or non-competitive inhibitors, which can alter the behavior of that enzyme to substrate action. So first of all, let's talk about competitive inhibition. So competitive inhibition is, yes, we have the enzyme here and we have the active site. And the active site usually attaches to that substrate. But in the environment, for some reason, there's something called a competitive inhibitor. And these competitive inhibitors can compete with the substrate for the spot of the active site, which basically means that the reaction can occur a lot slower if there's a competitive inhibitor, because that can have competitive inhibitor can block the behavior of the substrate, potentially reducing the product being produced. The other type of inhibition is called non-competitive inhibition. And yes, you can have an enzyme that attaches to a substrate normally as well, but it's competing for what's called a non-competitive inhibitor, which is a location on the enzyme, not the active site, known as the allosteric site. This is on the enzyme in a totally another location other than the active site. So say the active site is over here. You have on the other side the allosteric site, which is where the non-competitive inhibitor attaches to. What happens in this case is once the non-competitive inhibitor attaches to the appropriate allosteric site, that enzyme is now null and void, and that substrate cannot attach to that enzyme, which can be a problem and lower the functionality of that enzyme. So that's important to note. Enzymes can be functioned by the substrate concentration and these inhibitors. So they're competing for that spot. And this is more of an environmental issue that can also partake as well, that can affect how enzymes catalyze. There's something called cofactors, which we'll talk about later, but they also impact as well the enzyme efficiency and how they work properly. So let's try to tackle a quiz question. Take a moment, pause the video, and see what you come up with the answer when reviewing this question. Okay, so it says a chef is preparing for dough, and for bread, we know that there is bread that has some time that it needs to be created. So that's just some side information. And it says, according to the induced fit model or the enzyme activity that occurs, how does the enzyme in yeast, a type of protein, interact with sugar in the dough to ensure proper fermentation? Did you select the correct answer? If you selected C, that is the correct answer. Let's review all the answer choices here. With the answer choice A, it says the enzyme in yeast is rigid and only works if the sugar molecule perfectly fits into it. Remember, we talked about the lock and key method and how the lock and key method was a previous old version that promoted rigidity versus flexibility, and so that's why that's incorrect. With B, it says the enzyme in yeast slightly changes the shape of the sugar molecule to best fit the enzyme. And we know that the enzyme needs to adjust its shape, not the substrate itself. So that is not well for a proper choice. C says the opposite, which says that the enzyme in yeast adjusts, which we now know to be true compared to answer choice B. And then D says the enzyme in yeast permanently changes shape. And we know there's flexibility with that enzyme, so it's not going to permanently change. I hope this video helps understand the beginnings of enzymes. Make sure to hit that like button and subscribe.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.